Welcome to Almost Here, Round the Corner Future Technology podcasts with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcasts. I have Rosalyn Abbott. She's a professor at Carnegie Mellon University, and we're going to be talking about tissue engineering of fat. It's going to be very interesting. She runs the Abbott lab, and we're going to talk about her work there. So, Rosalyn, how you doing?
Good. Thanks for having me on the show.
Yeah. Well, tell me about the lab and what brought you to this path. What made you decide to study what you're studying, and then we'll go into more stuff from there.
Okay. Yeah, sounds good. So I started at CMU last August, so my lab's a little bit young, and we've been working on engineering fat in the lab, which might sound weird. I get asked a lot, don't we have enough fat? But actually a major challenge for designing drug therapeutics is that our current understanding of human disease mechanisms is based solely on animal models or really simplified cell culture models. In particular, fat or adipose tissue has long been thought of as this passive organ that's only purpose is to store energy or to insulate, protect the body. But recent work has demonstrated that it has more dynamic roles, releasing factors that affect other organs in healthy and diseased states, which is why obesity is really a high risk factor for so many other diseases. So more research is really required to determine how fat or adipose tissue is communicating with the rest of the body. We also need more studies to determine what factors cause those. Yeah.
What are some of the surprising things that you've learned about fat tissue that people have misconceptions about?
Other than the fact that it's this dynamic role releasing factors.
What have you learned about fat that a lot of people have misconceptions about? Again, most people think it's just tissue that sits in your body and stores energy, but what have you found that it does instead? What's different about it?
So really fat communicates with other tissues and it releases factors that communicate with those other tissues. So these factors can, for instance, be inflammation. So in the case of obesity, you're in a chronic state of inflammation that causes stress on the rest of your body. Other factors that we kind of look at are these things called obesogens, which a lot of people don't really know that word. It's these chemicals that actually predispose patients to gain excess weight. So we're exposed to them and they kind of alter our set point of where our metabolism is. And so our lab was one of the first to kind of show that BPS, which is very similar to PBA, which is a known obesogen found in food and drink packaging, is also an obesogen. So that's the one that they kind of substituted all these BPA-free plastics with. So it also has these kind of altered set points that people,
Wait, so BPA is bisphenol A, and that was found to be what, a carcinogen or a mutagen? And what was the problem with BPA? And then let's talk about BPS. But BPA, was it? What was the problem with BPA? I know that it was in baby products, and it was now it's not. But what was the problem with BPA, first of all?
Yeah, so BPA is they call it an endocrine disruptor. So it has large effects all over the body. What we look at is how it affects your metabolism, but it has a lot of other factors, that affect the babies at different developmental stages and how their neurological systems develop, and even adults at later stages too. So it's got a lot of really powerful effects on humans.
So bisphenol A now is changed to bisphenol S, or what does BPS stand for?
Yes. Yeah, so it's just a slightly different chemical analog of BPA, but so those are what most of the BPA-free packaging is made out of.
So does BPS disrupt the human body in the same way as BPA? You said it was called an obesogen. Was BPA an obesogen, or is this different?
So it does have similar effects. I don't know if it has all of the same effects as BPA because we've really just looked at it in the context of obesity and adipose tissue accumulation. But it does suggest that it might have similar effects.
So when you consume a drink in like a plastic package, I guess you ingest some of the BPS, and then what does it do to your body?
The problem with a lot of these chemicals is that they they're lipophilic, so they're actually predisposed to kind of accumulate in your adipose tissue. And so they kind of stick around for a while and can have effects. And what they do is they really act as hormone mimics, which hormones are one of the ways that our body uses to communicate endocrine disruptions. So basically, they kind of stick around and act like these hormones do. So they kind of change the way our metabolism works.
Oh, so all right. So they act in the same method as hormones. And what does BPS in particular do? Like, how does it cause obesity, or how does it predispose the body towards obesity?
Yeah, so it stimulates the cells to accumulate more lipids. So in the same caloric intake, a person that's had this exposure will accumulate more of the fat from it. So it doesn't necessarily cause obesity. It just makes you more predisposed to accumulate adipose tissue. So given the same kind of diet, then you'll be more likely to gain more weight.
Well, how does it predispose you? What have you learned about how the body decides whether to turn a food source into fat?
Yeah, so there's a lot of different regulatory mechanisms, and we know just a few of them actually. But basically, it's just kind of programming the cells to accumulate triglycerides, which is actually the molecule that is stored in your adipose tissue.
So for some reason it changed. You said the body has a set point. You alluded to that earlier. Can you talk more about that and how that affects people's weight and how diet affects them because of the set point? What happens?
Yeah, so I think we're still learning a lot about this set point, and there's a lot of different factors that affect it. So your genetics, for instance, whether you've been exposed to some of these environmental factors, and certainly other things as well. So I think we're still learning what actually causes this set point, and that's actually a big focus of what my lab does. So we create these tissue engineered models so that we can start looking at these factors and see if we can determine, did smoking affect how we accumulate adipose tissue? We think it does, but is that something that we can look at, or are there other factors? Whether you exercise a lot or other lifestyle factors as well. So it's kind of a bigger picture, and we're hoping that with using these tissue engineered models in the lab that we can actually start to figure out and tease out what this kind of combined picture looks like.
So what's the consequence of the set point? I'm interested. What will happen is the set point, let's say I eat a meal that would normally be, all stored as fats. Will the body, just because of a set point, at some point stop storing, a food source as fat? But it's a hard question to ask. What does the set point look like? Yeah, what does the set point look like in action? Everyone has a set point, I'm sure. So what happens to the body before it hits the set point, when it hits it, and then after it hits it? What goes on metabolically, do you think?
Yeah. So I think when I'm thinking about a set point, so I can give you kind of going back to the obesogen example. They exposed the mothers of mice to BPA or TBT. TBT is one of the bigger ones. That's a pesticide. And so the pups, they were exposed to the same diet. And the ones that had been exposed in utero, which is a developmental stage, or in the mother's womb, they gained more weight in response to the amount of calories that they took in. So they were given the same amount as the pups that were not exposed, and so they just accumulated more weight. And so that's what I'm kind of talking about with a set point. It's the ability to kind of accumulate more adipose tissue with the same caloric intake. And so it's not necessarily before the set point or at the set point or another. It's just kind of how your body responds to those calories. If that makes sense.
Well, what do you think? What feedback mechanism do you think lets the body know? Okay, we reached the energy state we want to reach. Now, change how you utilize a food source.
That's a great question. I don't know if we necessarily know how it kind of regulates that, and so I think that's something that we're trying to work a little bit more on.
Is there a way to empirically test and determine someone's set point? And what units would you express a set point in if you could find such a thing?
Probably energy, I would think. If I could express something like that. And how could you test for it? I guess it's harder to do in humans, but it's because you can't kind of well, I mean, I guess they have done some studies where they've looked at like forcing people to have the same amount of calories and what that kind of does to them. But it's not necessarily something that you would want to do ethically. Yeah, so I think that would kind of be the best way to determine it. But what we're hoping to do is, because we have these tissue engineered constructs that we have in the lab, that we could test it that way. And so you could get a small sample of adipose tissue and, try to determine its set points, try to determine what it responds to, because we all are very metabolically different. And so I've seen that in my cell cultures, and it's been something that at some point has been very frustrating, because it shows really variable results between different patients. And so, given the same kind of factors, they'll respond differently. And so I'm hoping that our models can be used to look at these kind of different factors and set points and things like that in the future.
When you have a tissue sample, how many cells is the sample, and have you been able to look at the behavior of individual cells, or do you have to look at them as a group, and therefore it's harder to figure out, what's going on?
Yeah, so you can look at them in smaller volumes, but then it's harder to extract a lot of data from them. So we usually do aggregates. We'll have in one, usually we do these kind of round scaffolds, 2 millimeters thick by 4 millimeters in diameter, little cylinders. And so in there, we'll probably have about 100,000 cells. And that's kind of what we use for our different assays or kind of measurement points. But you certainly can do some at the cellular level, but then you don't get as much data out of it.
Do you think that the fat cells are communicating with the other tissues in the body? I mean, I guess they are chemically, right?
Definitely.
They have receptors, and they send out signals once they take in an element of food. I mean, what mechanistically, what's going on when you consume food in your body? What are the fat cells doing? What are they getting?
Right. Yeah. So there are these different factors that are secreted that do work systemically. So they travel throughout your body, and some of them tell you that you're hungry, or some tell you that you're full. And so as you're eating, these different factors will say, oh, we've actually got enough calories, you're full now. And so that's just a molecule actually that your body will secrete, and it starts actually in the adipose tissue. And then over time, kind of as you're fasting, between meals, or even longer than that, your body starts burning these calories, and so other factors are secreted to know that you're hungry and that you should kind of eat something so that you can replace those calories. In the fat tissue, they're stored in triglycerides, and so the body knows that as it's eating, it should store in these adipocytes these triglycerides, and during these periods of fasting, it's a process called lipolysis, so that those triglycerides are broken down into free fatty acids, and those are kind of what power your muscles and everything else during these periods of fasting, I guess.
So what does it look? What does a fat cell look like? You call it an adipocyte or an adipocyte, right?
Yes. Yes, an adipocyte. Yeah, they're these big round cells. So they're actually almost 90% filled with lipids, are those triglycerides.
So like in the cytoplasm of the cell, you've got, I guess, the nucleus and the mitochondria and all the other stuff, but you're saying the vast majority of the cytoplasm has what, like globules of triglyceride in it, or what does it look like?
Yeah, it's they call it a unilocular lipid droplet because it's just one large lipid droplet. So if you can picture a cell being, a circle, and it does pretty much look like that, maybe more of an oval, then it's got this big round oval inside of it that takes up most of the cytoplasm, and that's the lipid droplet.
Weird. So I mean, what about the nucleus? The nucleus sits to the side of it, or it doesn't envelop the nucleus?
Yeah, it sits to the side of it. Yeah, it's more like kind of like a crescent almost.
Well, that's weird because
it's a pretty funny looking cell.
Well, that would suggest that within the globule that it has its own microenvironments. I mean, doesn't it affect how things move about in the cytoplasm? And let's say it takes up a large percentage of the volume inside the cell. I mean, the cell membrane that abuts that area, I would think, would be affected, and the transport of things within the cytoplasm, and again, it would create its own microenvironment.
Oh, definitely. Yeah, for sure. Yeah, it's a very unique environment. And that's why I was talking about some of these molecules that are more lipophilic, which just kind of means, they like kind of being in the fat, so you've got all these fat cells and you've got these chemicals that are kind of happy to be there.
Well, have you opened up any of these globules, and does it have is it uniform throughout the volume of the globule, or does it have a structure to it? Are there different things present in the outside of it or the inside of it, the center?
Yeah, I haven't myself, but other people have. They've looked at the proteins that surround the lipid droplet, and yeah, there's very specific proteins that kind of keep it intact and regulate it so that it can, change and secrete, those, or go through that process of lipolysis where the free fatty acids are secreted. Yeah, so it's tightly regulated for sure.
Well, what happens in lipolysis is like a hunk of the globule, I don't know, put out through the cell wall, or is it changed within the cell itself into fatty acids? Like what happens?
Yeah. So it breaks down into free fatty acids and glycerol, and so both of those are actually secreted. So yeah, it basically takes that triglyceride and kind of takes the backbone of it, and yeah, so it's got these two new proteins that are then secreted and can go other places in the body and provide energy.
So do you consume the entire globule, or parts of it be consumed?
No, just parts of it would be. Yeah, so it would still remain. Yeah, actually, adipocytes can last for up to 10 years in the body, so they're real slow turnover as opposed to some other cells that can, last only a few days, or they stick around.
That's interesting. What's the consequence of them then, that adipocytes are so long-lived and they have, this triglyceride globules stored inside of them? I mean, anything that jumps out at you that you find is interesting or different?
Yeah, so, we talk about kind of these obesogens or these other chemicals that, are eliciting changes. And so you can imagine if they're persistent and they're sticking around these cells that are also persistent, then their actions can kind of occur more long term. So that's still research that we're looking into, but I have a feeling that that's kind of related. This kind of memory almost of these exposures.
Yeah, so once if you have an adipocyte, and let's say most or all of its triglyceride globule is used up, you're saying that you believe it has a memory that it will tend to reaccumulate preferentially a similar size globule, or what will it do? What's the memory component to?
So just that when I was talking about memory, I meant, like the chemical would kind of be there as the memory of the cell. But yeah, I do think they are all about the same size. But that obviously changes with caloric intake as well. So when we get these tissues and we look at the cells, they are pretty uniform. I mean, there's obviously variation, but of how they've accumulated lipids. But that will definitely be really dynamic as the person maybe gains or loses weight. The cell size will also change.
Do these cells tend to cycle their triglyceride globules, or they sit there sometimes for months or years, the same substance never moving?
I think they cycle them. It's a pretty dynamic process, and the cells do move around too. They're not just kind of sitting in one spot as well. So yeah, it's a dynamic organ for sure.
Yeah, it's just so odd. That's why I'm asking so many questions, because I never really thought about it before. But now that I do, it's just a very strange thing,
Yes, it is. It's a really interesting organ system for sure. I think it's kind of understudied, and so that's why I think it's important to do a lot of this work.
You're saying you refer to it as an organ system. So a group of adipocytes, I guess they communicate with each other and they form an organ system? They're not just independent cells sitting there?
Definitely. Yeah, so we talk about the adipocytes, but there's also other cells there. I'm sure you've heard of adipose-derived stem cells. So those are kind of mixed in between all of these like larger mature adipocytes, and they're usually next to what we call the stromal vascular fraction. So the blood supply basically that is providing kind of this communication with the rest of the body. So you've got stem cells and you've got neurons right there. So it's innervated. You've got other cells that are kind of supporting that vasculature that are more like muscle cells, and then of course you've got the vasculature. So yeah, it's this organ system that's got both adipocytes and then also that blood supply as well.
So what do you see as the goals of this organ, the fat organ? What does it do? I've heard it like produces estrogen and other substances in the body. Like, what have you observed it to do, and how does it act in concert with itself?
Yeah, so I guess what we look at is trying to use it to study the transition of obesity to type 2 diabetes. And so how this expansion in this adipose tissue is related to these large systemic changes in the body and, other diseases for sure. It has big effects on, obesity has big effects on like cardiovascular disease and even some cancers. So we kind of look at how, as this is expanding and changing, what factors are being secreted, what's happening in order to cause all these other changes that occur throughout your body. And, we talked about those chemical communications. So what is it communicating with the rest of the body that's causing all these problems? And one of the ways that we think it has that effect is through inflammation. And so that's kind of what we've been looking at in the lab, as opposed to kind of these other chemical exposures, to see if we can kind of figure out a target for maybe some better anti-diabetic drugs, or trying to make people more insulin responsive.
So what have you noticed when someone gains weight? What happens? Are more adipocytes created from the adipocytic stem cells, or do the existing ones seem to just take on more triglyceride until they hit like a size threshold, or what happens?
Yeah. So it's both, actually. Yeah, you are absolutely right. So more of those stem cells are recruited, and then also the size of the cells increases. So both of these factors actually cause the body to then become this chronic inflamed state that actually lasts the entire time you're obese, and it is reversible if you lose weight. So that's probably, and that's what most people think is really related to this change into this more insulin-resistant diabetic phenotype, type 2 diabetes.
Well, what happens if you deplete a given set of population of adipocytes of their triglyceride globules? Do they ever deplete to nothing, or they only get to a certain small size that stops? And what happens then?
Yeah, they can completely empty. Yeah, and then they kind of go back to the state of the stem cells if they were totally depleted. Yeah. And we see that in the lab. If you take adipocytes and you do something called a ceiling culture, so they kind of float to the top and stick to the top of the flask, they'll kind of deflate and lose all their lipids, and then they look a lot like stem cells, and they behave a lot like stem cells too.
You said some of them last for years. Have you seen any correlation between the amount of triglyceride that a population of cells holds and the length of time in which it'll keep itself alive?
Oh, interesting. I have not seen anything related to that, but that's a very good question. I would imagine, that probably is somewhat related. But yeah, I wouldn't be able to tell you for sure.
So you have populations of these adipocytes, and what kind of experiments are you running? Are you stressing them chemically? Are you what are you doing to cause them to evoke different behaviors?
Yeah, so we do definitely stress them chemically. So there's inflammatory factors that we can add. You can add inflammatory cells themselves to the cultures as well. You can just kind of look at tracking a whole bunch of different patients and seeing how they respond to different factors, which is something that we're working on. And then you can treat them with the different factors that you suspect are going to be related to these obesogens, because right now it's a really young field, and it's only been since 2006, I think, that people have even started to look at these. So there's a lot to be learned, I think.
Have you had let's say two populations that are spatially segregated? I'm sure in their world, an inch is a lot, but they're the same solution. And have you tried to stress one of the populations and see if the other reacts and changes?
Oh, interesting.
See if there's communication.
No, we haven't done that kind of experiment, but sometimes you can do experiments we've done with different tissue systems. So having the fat and, putting some other tissue on top of it or in the same well, and then seeing what kind of factors are being communicated between them. That's definitely something that we're interested in working on. We've actually started creating these perfusion systems that we're hoping to integrate with some other collaborators to try to get more of these systemic effects. So whether it's, the liver or the pancreas, which are also involved in type 2 diabetes, and see how the adipose tissue is communicating with these other two, organ systems as well, is something that we're working on.
Yeah, and are there different types of adipocytes? I'm overweight, for instance, so the fat I have in my stomach or around my stomach, is that different from the fat in my leg or in my upper torso, my arm?
Yeah.
How is it different?
Yes. Right. Yeah, there are definitely different types of fat. There's visceral, which is actually the fat that's kind of more around your organ systems, and those are more associated with diabetes and inflammatory responses. Then you've got subcutaneous would be kind of your belly, which is actually where we get most of our tissue because we get tummy tucks and liposuctions. Then there's mammary adipose tissue, which is entirely different as well. And then there's even fat that's kind of in your bone marrow, and that behaves a lot differently. So there are all these kind of different sections of adipose tissue that do respond very differently. So yeah, they've got their own kind of types. Yep. Oh, and then there's a type called brown fat. Oh, sorry, go ahead.
Oh yeah, tell me a little bit about brown fat. Go ahead.
Yeah, so brown fat's a kind of interesting one because instead of kind of using the energy to kind of help the body, it burns the fat and creates heat. So it's really prevalent, in like infants, so they can have this kind of non-shivering thermogenesis is what it's called. And so we have really small amounts of it in adults, but people in kind of colder climates will have more brown fat. So it's a completely different kind of adipose tissue.
Yeah, I've read about cold exposure possibly could help increase the amount of brown fat in your body.
Yeah. I think it's been a promising way people think of kind of losing weight kind of quickly.
Do you have any insight into? Yeah, do you have any insight into what happens when someone gets liposuction, for instance? I've heard it's like, it's not a very safe procedure. It's like, any insight into what's happening when someone gets that?
Well, so yeah, I haven't studied anything related to this, but I've read a little bit about it, and it's interesting because it does decrease the weight really quickly, and it kind of changes this whole microenvironment that we've been talking about really fast, right? It kind of takes it away, and so how the body adapts to that could be good, could be bad. In a lot of cases, these liposuctions actually do help because they decrease the weight, and they've got a lot of extra fat other places that seems to be okay. So I think I wouldn't necessarily say it's a bad thing. I just think we don't understand a lot of how the body actually recovers from that, and whether there are maybe certain people that would recover a lot better than others, for instance. I'm sure that there are kind of some factors that we might be able to figure out that might predict that really well, but I don't think we're there yet.
And I guess last thing, I'm just, all these ideas coming into my head, but I've talked to a number of companies that are creating organoids. They're like pseudo organs. So I don't know if that would be something where you would couple fat tissues. Excuse me one second.
Yeah. Go ahead.
Okay, sorry about that. I'll repeat. So I've spoken to a number of companies that are developing organoids, heart organoids, kidney, etc. Would that be something that you'd want to do, where you take an organoid and put fat tissue around it and see how it interacts differently with different organs of the body, even if they're organoids? I don't know if that would be helpful, but just an idea that I had.
Yeah. So I think the best way to kind of describe what we're doing is creating organoids. And so, that's the goal, is to hopefully integrate these with different tissue systems. I mentioned like the liver and the pancreas, but certainly there are other tissue systems where there's this communication that would be really interesting. And I think that's the way that we're going to really learn how these tissues are communicating and figure out ways of kind of altering that communication when it is going down a disease state. I think that's a big direction of where we'd like to go, and I think it's a really important one.
Well, very good. So what, last question. So what are some, you probably covered it, but any other surprising facts that you've learned about studying this fat tissue?
I think, I guess I've probably talked a lot about it, but I think just really what fascinates me is how the highly variable kind of metabolic status that people have, and trying to figure out really what causes those changes and how we can use that to, make people live healthier lives and have more predictable metabolism. So I guess, that's kind of what I think is exciting, and I'm hoping that we'll make some of those insights in the near future.
All right, well, very good. Well, what's the best way for people to, either contact you or the lab to find out more, to suggest collaboration, et cetera?
Yeah, so they can reach me by email, probably is the best way, and then we could set up a time to chat. So my email is rabbott, RABBOTT, and that's at andrew.cmu.edu. I'd be happy to talk to anyone who's interested in learning more.
That's great. Well, Rosalyn, I really appreciate you being on the podcast. It's been great. Thank you.
Yeah, thank you so much for having me.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, post a review to discover more future technologies that are poised to transform our lives for better or worse. such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.